Synthesis of deoxyribonucleotides. The enzyme is ribonucleotide reductase, which is specific for the reduction of nucleoside diphosphate. Deoxyribonucleotides are the building block of DNA, since the deoxyribose sugar is unique for DNA molecule. So adenosine diphosphate will convert it to deoxyadenosine diphosphate. Guanosine diphosphate converted to deoxyguanosine diphosphate. Cytidine diphosphate converted to deoxycytidine diphosphate, and uridine diphosphate converted to deoxyuridine diphosphate. This slide shows the synthesis of deoxyribonucleotide, which are the building block of DNA. And we can write this reaction as an equation. Ribonucleoside diphosphate by ribonucleotide reductase converted to deoxyribonucleoside diphosphate. This enzyme needs thyrodoxine, which is a protein contains 2-thiol group, which act as a hydrogen donor in order to react with oxygen from ribose to form water, and converted from reduced form to the thyrodoxine oxidized form. Then this oxidized form should be returned to reduced form, and this needs thyrodoxine reductase and not pH. The conversion of ribonucleotides to deoxyribonucleotides occur at the level of nucleoside diphosphate. All 4 nucleotides, ADP, CDP, GDP, and UDP, are reduced by the same enzyme, ribonucleotide reductase, and this requires NAD pH. Then all the deoxyadenosine diphosphate, deoxyguanosine diphosphate, deoxycytidine diphosphate, and deoxyuridine diphosphate converted to deoxytriphosphate obtained by ATP-dependent phosphorylation by nucleotide diphosphate kinase. So in order to prevent the incorporation of uracil into DNA, the deoxyuridine diphosphate dephosphorylated to deoxyuridine monophosphate by phosphatase enzyme. Then deoxyuridine monophosphate converted to deoxythymidine monophosphate by thymidylate synthase enzyme. Then deoxythymidine monophosphate is phosphorylated twice to produce deoxythymidine triphosphate. This slide shows the conversion of nucleoside diphosphate of all 4 bases. It means adenosine diphosphate, guanosine diphosphate, cytidine diphosphate, and uridine diphosphate to the deoxynucleoside diphosphate by ribonucleotide reductase enzyme, as explained previously. The most important point was deoxyuridine diphosphate dephosphorylated by phosphatase enzyme and converted to deoxyuridine monophosphate in order to enter the reaction catalyzed by thymidylate synthase and converted to deoxythymidine monophosphate, which phosphorylated twice to form deoxythymidine triphosphate. Then all these deoxynucleoside triphosphate will use as a building block of DNA, the reaction catalyzed by DNA polymerase enzyme. The synthesis of thymidine monophosphate from deoxyuridine monophosphate is catalyzed by thymidylase synthase enzyme using N5-N10 methylene tetrahydrofolate as the source of the methyl group. It means as a coenzyme. Synthetic inhibitors of purine synthesis. It means a drug. For example, sulfonamides, which inhibit the growth of rapidly dividing microorganisms without interfering with the human cell function. A sulfonamide interferes with the ability of bacteria to use folic acid to grow by stopping the metabolic process. Therefore, the bacteria are unable to reproduce. This slide shows the biosynthetic pathways of tetrahydrofolic acid, which is essential coenzyme and a sort of methyl group when converted to N5-N10 methylene tetrahydrofolate, and as explained previously, used as a coenzyme as a source of methyl group in order to form the thymidine monophosphate, and then this thymidine monophosphate converted twice phosphorylated twice in order to convert it into thymidine triphosphate and using as a building block for DNA synthesis. So by these steps, most important things in this biosynthetic pathway is one enzyme, dihydropyruvate synthetase enzyme and dihydrofolate reductase enzyme. Sulfonamide antibiotic, it means antibacterial, will inhibit dihydropyruvate synthetase enzyme. It means يكون in competition with para-amino benzoic acid, يعني competitive inhibitor to stop synthesis of folic acid when only بالmicroorganism, not in a human, do not interfere with the human biosynthetic pathways. It's only with microorganisms. It's a very important point. Also, trimethoprim will inhibit dihydrofolate reductase. It means prevent formation of tetrahydrofolic acid only in microorganism, not interfere with the human biosynthesis pathway. This is most important point. Other purine synthesis inhibitors, such as structural analogs of folic acid. For example, methotrexate drug used to control the spread of cancer by interfering with the synthesis of nucleotides and therefore interfere with the synthesis of DNA and RNA. Methotrexate is a drug that inhibits dihydrofolate reductase. An enzyme reduces dihydrofolate to tetrahydrofolate. So it decreases the supply of tetrahydrofolate. بالنتيجة شنو يصير عندي decrease بالsupply of N5-N10 methylene tetrahydrofolate, that is used as a source of methyl group and coenzyme for thymidylate synthase enzyme. So راح شنو interfere with the synthesis of thymidine monophosphate and thymidine triphosphate, which is the most important building block, especially for DNA. And also to affect all rapidly growing cell, not only the cancer cell, كل rapidly growing cell، فإذا هي toxic بالنسبة للهيومن. صحيح نستعملها بالتريتمنت للكانسر، بس الكانسر الديفينيشن مالته هو abnormal or rapidly growing cell. فهنا راح كل rapidly growing cell من ضمنها مثلا الهير cell هم راح يصير بيها interfere with synthesis من DNA، فيكون لها toxic effect. هاي نقطة مهمة ننتبه عليها. So these folate analogs not only inhibit purine synthesis, but by prevention or preventing methylation of deoxyuridine monophosphate to deoxythymidine monophosphate. So it lowers the cellular concentration of deoxythymidine monophosphate, which essential component of DNA. طبعا after انه اذا ما تكون هذا ما راح يتكون الثايميدين ترايفوسفيت وبالنتيجة ما عندي DNA. While trimethoprim, another folate analogs, but potent antibacterial activity because of its selective inhibition of bacterial dihydrofolate reductase. And it's very important point. Be careful when read this lecture.